Oxford. I won't characterize what his degrees are in because Dan is, in my opinion, part of a new generation of trainee where you are working at interdisciplinary boundaries. It's very hard to say what you want. He maintains an interest in the notion of subjective confidence and the physical signals that underlie that and the way that these things are manipulated and perturbed by social context. In other words, the way that we gain a feeling of knowing that we know something and how that compiles in with interactions with other people or in the presence of other people. So you have an obvious connection to things I'm interested in. And I hope at the end of this, all of you heard that I'm going to interrupt him during the talk, which we have a big effort in collaboration with, I think, a beautiful behavioral design that we'll hear about today that Dan and Steve Fleming have worked up and published most recently in Proceedings of the National Academy, and married to a set of techniques developed by myself and King Kishida at recording dopamine and serotonin and sub-second rates in human beings. So these are people doing this really interesting behavioral task while we look at dopamine and serotonin fluctuations in their brains, and we were going over the data today, and it's a really exciting start. Those methods are going to move out to wires pretty soon, and I think we'll become, clearinghouse for early cognitive experiments where we have real fluctuating neurobiology at a scale that fMRI was never going to give us. It's going to give us a different set of information. So with that, we welcome Dan.
So thank you, Rick, for organizing this and for helping me out along the way coming here. So the title is Neurocomputational Mechanisms of Private and Public Confidence in Humans. I'll first talk about the private part and then move on to the public part with the human multimetry somewhere in the middle between the two. So what do I mean by confidence? The confidence has many meanings in everyday language, but in decision neuroscience, we use confidence to refer to an internal estimate of the probability that a decision or a belief is correct. And here you should understand decision and belief in a very broad sense. So this is ranging from sensory perception to general propositions, for example, the proposition that I'm a good husband evaluated over a much longer time scale. And this definition of confidence might at first hand seem a bit esoteric, but as will become clear, we can also relate it back to everyday situations. Now, having a sense of confidence is, from a computational point of view, an incredibly useful signal for guiding your behavior and cognition in an adaptive manner. So, for example, when you have to make a series of decisions, and success requires that each decision is correct, imagine yourself assembling an IKEA furniture, or when deciding how much time to invest into learning, for example, preparing for a difficult exam. Now, confidence also allows us to recognize mistakes and rectify these before it's too late. And it might also allow us to change our mind as new information comes to light, something that Steve, my supervisor, has worked on. Now, public displays of confidence also seem to be important for social success. And here I've just done a quick Amazon search for book titles containing the word confidence. So you have Fake It with Confidence and Confidence in Social Magnetism. Now, this public dimension of confidence has received much less attention than the private dimension, perhaps because it's harder to study or quantify in the laboratory. And today I'll try to show you how we do that. So I'll take up this challenge and present my work on both the private and public sides or features of confidence, with a particular focus on the interplay between the two dimensions. So first, I'll describe how we study confidence in the laboratory, and then I'll give some highlights from the animal and human literature on the neural basis of confidence. Then I'll move on and present our work on private confidence, where we have used a novel task to isolate signatures of confidence in human fMRI and multimetry data that we just talked about. I'll then present our ongoing work on public confidence, focusing on how the mapping from our private confidence to our public confidence changes with the social context. And then finally, wrapping up, I'll consider the broader role of private-public mappings in human social function and dysfunction. Some of these terms will become clearer as they go along. Okay, so how do we study confidence? Now, this is a busy slide, but this is roughly how we do it. So a commonly used system for studying confidence, laboratory system for studying confidence, is sensory psychophysics. because we can use it with both humans and animals. We can't do that for more complex decisions, such as should I buy a car, for example. So typically on a trial, subjects would make two decisions. We have the first decision, which is the choice, which will be about some stimulus. This is the first order judgment. And the stimulus could be a cloud of dots, and the subject could have to decide whether the dots are moving left or right. It could be about a contrast grating, whether it's tilted left or right, counterclockwise or clockwise relative to vertical. Or it could be about an odor compound, whether it contains more of odor A than B. Now, after they've made a choice about the stimulus, they're then required to make a second order judgment, if you like. And this judgment, and in order to maximize reward over the experiment, this judgment has to somehow relate to the probability that the initial choice was correct. So what we do in humans is that we can elicit such judgments explicitly. So we can actually ask them, how confident do you feel? Although Reid says he can't exactly put numbers on how confident you feel, but at least it gives us some information into what people might be feeling. And in animals, we might instead ask them, so we can't ask them, so we use implicit measures. So one commonly used measure is waiting time. So imagine you make a choice. That choice might be correct or incorrect. Now, how long you're willing to wait for a potential reward should depend on how likely that choice was to be correct. Another used technique in monkeys is the opt-out paradigm. So a monkey is offered a sure but small reward. They also have the option to gamble on their choice, and then they would potentially receive a larger reward, but they might receive nothing in case the choice turns out to be wrong. So these are, so to speak, explicit and implicit measures of confidence, if you like. What we then do is to use data collected using this approach to characterize the cognitive processes or computations supporting confidence-based behaviors. And then in turn, we can ask how are these computations or quantities and variables instantiated, implemented in the brain. And there's sort of roughly two approaches to studying the neural basis of confidence. So this is the variable that we're interested in. We're interested in this internal estimate of probability correct. Now it's ultimately an unobservable variable, so we have to make inferences about it. And there are two ways of going about this. So the first one is a bottom-up approach. So what we do here is that we manipulate the stimulus, and then we look at them at the effects on choices and confidence-based behavior upstream. So for example, we might manipulate the stimulus experimentally, varying the strength of the sensory evidence, or we might rely on inherent stochasticity in sensory processing. But in a nutshell, the basic idea shown on the right here is that evidence strength will be correlated with probability correct. We can therefore use evidence strength as a proxy for confidence when we go and analyze the neural data. That's the idea. Another approach is to simply take these reports and then regress these back onto neural activity. And the idea is that the higher reports, whether it's an implicit measures obtained using implicit measures such as willingness to wait, or explicit measures such as using a confidence scale, the idea is that higher reports are associated with higher probabilities of being correct, and we can therefore use reports as a proxy for confidence. Now, people have used these approaches with various species and techniques to identify neural areas that are involved in, or at least predict confidence-based behaviors. And the greatest hits collection include rat orbitofrontal cortex, monkey LIP, monkey midbrain dopaminergic neurons. And then in humans, we find striatum, dorsal anterior cingulate cortex, VMPFC, and DLPFC. So just looking at this, it seems that the whole brain is encoding confidence. Now, this can't be right. Presumably, what we're looking at, what we might be confounding or confusing different kinds of computations and quantities. And a key problem is that the standard approach confounds confidence, probability correct, with related but distinct variables or computations. So under the first approach I showed to you on the left, it's difficult to dissociate probability correct and evidence strength. However, evidence strength is only one component of a confidence computation, as I'll show to you. And under the second approach, variation in observed confidence-based behaviors could be due to a whole range of different factors, some of which only partially related to probability correct. This might be risk aversion or individual report biases. Okay, so I've told you how we study confidence in a laboratory setting. I've introduced you to the idea of using sensory psychophysics for studying confidence. Then I've touched upon the rather confusing literature on the neuroanatomical basis for confidence. So now I'm going to talk you through the private confidence part. Okay, so we have a problem in confidence here indicated by the blue-yellow color scale can then be inferred from a single stimulus dimension. And what we did to solve this problem was to create a task in which confidence must be constructed by taking into account not only one, but two stimulus dimensions. And it might seem simple, but it does the trick, as I'll show to you. And in this case, confidence must be constructed, as indicated by the color map, must be constructed by integrating multiple stimulus dimensions. And the idea is that confidence therefore has to be represented or computed separately from any given stimulus dimension. So we're trying to tease apart this internal estimate of confidence, probability correct, from the different factors that go into that computation. So how did we do that? So we used a sensory task, what's known as the random dot motion task. So in each trial, subjects are presented with a random dot motion stimulus where there's an underlying direction of motion, and this direction of motion is sampled along the full circle. So in any given trial, it could be in any direction along that full circle. And what we do is that we vary the coherence, and that means that we vary the fraction of dots that move in the specified direction. The rest of the dots move around randomly. So when the stimulus is of low coherence, then there are only a few signal dots, and it can be hard to form a reliable sensory percept of the task. However, when coherence is high, you get a much more reliable estimate of the motion direction.
How do you deal with the impact of what a visual psychophysics person would call attentional load, like the total number of dots? How do you choose that in which you then take different fractions out of coherence?
No, so the total number of dots remain the same, but it's the relative fraction of signal and noise dots.
How did you pick that total?
How I picked that total number of dots, previous literature.
Okay.
But yeah, we can discuss that. But yeah, it's a good point.
For example, it's quicker when there are a lot of dots, or maybe a few. There's sweet spots for that, depending on the fractions you're using.
So there are a lot of different features that go into the construction of that stimulus. So there's the speed at which they move, there's the size of the aperture, there's the total number of dots, and all these features in a much more lower level sense impact on the reliability of your sensory percept.
Yeah. Let me just for clarity ask a question.
Yeah.
Your first part, which was, it's kind of a confused literature for multiple reasons. You didn't state them all. But it seems to me, given what you're going to do and what your paper does, that you wanted to dissociate the difference between the difficulty of the perceptual problem and some readout of how well you feel about your answer.
Yeah.
Okay. And a lot of the previous work had conflated making hard perceptual problems made you feel less confident, and easy perceptual problems made you feel more confident, but that's just
I think so this is a very important point. So in the monkey literature, often you use this random dot motion task. And if you're interested in confidence, what you do is that you just vary the coherence. And then you see whether monkeys feel more confident, whether are they more likely to make risky choices when coherence is high. But as Reed said, that the problem is that it's quite hard to tell apart whether it's confidence that you're then looking at when you go and record in the brain, or whether it's a representation of the perceptual difficulty, if you like. So this is one feature that we vary. So I'll refer to this one as coherence. And the idea is that it sort of targets sensory reliability. So subjects are then, after they've seen the stimulus, they're then presented with a reference. And what they have to do is that they have to say whether the direction of motion, the perceived direction of motion, was to the right or to the left of the reference point. So was it towards the orange or the blue part? And then this reference can vary from trial to trial. So there are these two different moving parts to the task. So one is the coherence, as I just introduced you to, and the other one is the reference. And in this manipulation, I'm going to call that distance. The idea is that it sort of targets boundary choice difficulty. And we can have small distances and large distances. So imagine you might see the direction of motion pretty clearly because it's a high coherence stimulus. However, I now give you a very difficult reference. So the judgment suddenly becomes much harder, even though you saw that stimulus fairly reliably. So subjects make a choice, and then after they've made a choice, they're asked to estimate how likely do you think you were to be correct on this trial. Okay, so just to recap, so we have these two dimensions, coherence and distance, and as Reid mentioned, the problem is that in the standard literature is that probability correctness is directly correlated with sensory reliability. But what we do by introducing this reference manipulation, this variable reference, is that we tease apart probability correct from sensory reliability and vice versa and the same for probability correct and boundary difficulty through the manipulation of coherence. So what do the results look like? They look like this. What we're looking at here is choice performance, so choice accuracy. On the x-axis, we have the distance manipulation from very difficult to very easy distances, so references, if you like. And then the color denotes coherence. So blue is low coherence, red is high coherence. And as you can see, choice performance increases with distance, but it does so more steeply for high coherence. So your objective performance is a function of both factors. And if we look at subjective reports on the right, what we see is that subjective reports also track both of these factors. So confidence increases with distance, and it does so more steeply for high coherence. Now, interestingly, as you can see here, coherence has a higher impact on subjective experience, even though distance has a higher impact on choice performance. So it's perhaps because it's a more salient feature of the stimulus. Okay, so we're now going to leverage our task to search for a neural basis of confidence. And what we'll do is first to use our experimental task, our experimental factors, to identify candidate neural substrates. And then we'll ask whether this substrate or these substrates can predict variation in subjective reports. So it's sort of a two-step procedure. So what I showed to you here are the results of a whole brain search for neural areas that encodes coherence, distance, as well as their interaction. So what we asked here is, which areas care about all of these different components? And what we find is that the only area that tracks coherence, distance, and their interaction, all three features, is the PGACC, which sits in the medial wall of prefrontal cortex. And this is just to back up that claim. So these are our eye analyses. On the left, you see across subjects, you see the contrast estimates for each of the three factors, coherence C, distance D, and then their interaction. And you can see that in PGACC, at least in terms of the contrast estimates, it seems to encode all three components. And then on the right, what we're looking at are activity time courses that have been upsampled. So the dotted lines indicate the onset of the stimulus and the reference. And you can see that the PGACC activity time course also encodes all three factors. Even so, when we control for reaction time, shown in blue.
Wait. So in this graph at the right,
Yeah.
first vertical dotted line is when the dots start moving.
Yeah.
The second line is when the reference line comes out, and you're asking me, is it
To the left or to the right?
Is
Yeah.
it to the left or the right of that?
Yeah.
Okay.
Exactly.
So that's a 1 second delay.
Yeah.
So do the dots go out when the reference line comes on?
Yeah.
Okay. How long is the reference line illuminated?
Until you make a choice.
Oh, so reference line comes on and the dots extinguish.
Yeah.
So you still know where the reference line is, no ambiguity there.
Yeah.
You're having to remember.
But you're having to sample from some visual short-term memory. Yeah.
And then what's the delay between that? What's the average delay between that when they answer?
So if we looked at reaction time before, I didn't show it to you.
Showing that here? Is that what RT is here?
No. Yes. So I'm not showing. I didn't show you reaction time, but it's about 1.5 seconds.
You're showing me the betas and the ROI.
So these are the
RT.
So these are the betas in the RI for RT. So the faster the response, the higher the activity. But the reason why I'm throwing in reaction time here is that reaction time is often used as a proxy for confidence. If that makes sense. So we're just trying to control for it. Yeah.
Just as a complaint about the former work, reaction time is a bad proxy for that because animals temporarily discount things. And so your willingness to wait is conflated with your evaluation of the near-term future.
Yeah, yeah. So reaction time
And your reaction time is
reflects a whole bunch of different factors. Yeah, exactly. Okay, so we have identified this area, PGACCs, and now we're going to try to see if PGACC activity predicts subjective confidence. And what we have the reports that people made in the scanner. So they made a report about every 5 to 10 trials in the scanner. And what we also do is that we create a computational model of what they would have said had we probed them. And the computational model is built using pre-scanner data. So they're trained extensively on the task where we elicit confidence in every trial. And then we use this data to create this model of trial-by-trial confidence reports. So very briefly, so it's in a regression framework, and then we fit the parameters using pre-scan data, and then we feed it the scanner data to as you see on the right, to create some probability distribution of how likely they are to make a given response, which gives us a proxy of what they would have said on each trial. And what we show here is just that the PGACC tracks confidence. So at the top, what we're looking at is single trial activity estimates of PGACC activity. We've binned it from low to high using 6 bins. And then we look at how high is confidence in each of these bins. And you can see you have an increasing slope in both of them. And at the bottom, we're looking at the beta, if you like, predicting confidence as it's encoded or unfolds in PGACC activity. Okay, we can also look at sensory reliability. And here we're just looking at a main effect of coherence, ignoring distance and the interaction between coherence and distance. So it's kind of like an exclusive masking approach. So we're only interested in areas that only care about coherence in the fMRI data. And what we see here is that we see activity in the striatum, more posteriorly in area MT, which makes sense in the IPS, which might overlap with what you might call LLP monkeys. And this is interesting because the striatum and IPS have previously been implicated in encoding probability, correct? But at least in this case, in the human fMRI data, it seems to only reflect sensory reliability. We can also do the same for boundary difficulty, main effect of distance. And here we see singlet activity. And again, the singlet pre-SMA DACC has been implicated in encoding probability, correct? But in our task, it seems only to care about the boundary difficulty.
What's the difference in the blue and the yellow?
Positive and negative.
Okay.
So the blue is negative.
These are corrected
Yeah.
maps?
Yeah.
Okay.
So these are whole brain family-wise error-corrected maps. 32 subjects. Okay. So I've shown that PGACC tracks confidence in a local manner. So it seems to track your confidence in your here and now choices. And I think it's interesting that a recent study by Mark Whitman from Matthew Rushworth's group in Oxford showed that PGACC also seems to track a global estimate of confidence. So they created a task in which in which the feedback was rigged. So you kind of had to track feedback in order to figure out whether you were doing well or poorly on the task. And what they found is that the PGACC tracked this running average across over feedback back into time. So it seems that they're both these short and long time scales in the PGACC. Okay, so now I'm going to change gear and share some of the exciting work that we've been doing, Steve and I have been doing with Reed's team here in Roanoke and with Ken Kashid at Wake Forest. And what we do here is that we acquire voltammetric measurements of neurotransmitter release in human striatum, doing behavior on the psychophysical task I just showed to you. So why do we think that there's a link between neurotransmitter levels and confidence? We know that inappropriate neuromodulation is involved in a range of psychiatric conditions, such as anxiety and mood disorders, and that these conditions are associated with altered confidence levels. And this is some of the recent work coming from Steve's lab. And there's also evidence from the animal literature that dopamine and serotonin are involved in signaling predictions and uncertainty on various learning and decision tasks. So there are different reasons why we might think that there is a link between neurotransmitter levels and confidence. So how does this work? So this is just a cartoon overview of how the voltammetry protocol work. So what we do is that we acquire voltammetric recordings from the striatum in patients with Parkinson's disease or essential tremor while they perform our task in the surgical suite. And we then use machine learning algorithms or a machine learning approach trained on in vitro data with known concentrations of serotonin and dopamine. So we train it on data where we know what are the dopamine and serotonin levels. Then we use the algorithm, if you like, to deconvolve the voltammetry data into time series. And what we get is that we get time series for dopamine and serotonin sampled roughly 10 times a second, so about every 100 milliseconds.
By the way, that first paper is +1.
Oh yeah, +1.
Just FYI.
Should have been plus biology. Yeah. So I'm not. This is all I'm gonna say. And then the 3 papers at the, and I could really invite you to read the 3 papers at the bottom where Ken and Reed and the rest of the team outline the procedure, sort of starting from building the electrode and then using it in these cognitive tasks. Like really cool work. So this is work in progress, and we now have data from 6 patients, but what I'm going to show you here is behavioral data from 4 patients and neural data for 3 patients. For logistic reasons. So just to recap, remind you, so we have these 2 factors. We are varying coherence, sensory reliability, and we're also varying distance, the difference between the motion direction and the reference, okay? And we're first going to look at the behavioral results. So at the top, we have data from a pre-surgery session where we calibrated the stimulus features, and at the bottom, we have data from the surgery session. And on the left, we have choice performance, and on the right, we have subjective confidence. I think, first of all, what's striking is how well the task works in the patients, both outside the surgical suite and inside the surgical suite. So as you can see, if we look at accuracy, on the left, the distance has an impact on choice performance, more so for high coherence. So this is what we saw before in our PNAS paper. The data for confidence is more subtle. It definitely doesn't go against what we might expect. It's in line with what we would expect. So there's some separation between high and low coherence, and confidence seems to increase with distance. So remember, these are people with movement disorders. So there's a substantial amount of effort involved in moving the marker around on the scale. And we're looking into different ways of taking that into account. Okay, so the neural data. So what we're looking at here are neurotransmitter levels for dopamine and serotonin across a trial. So 0 is 1 second before the stimulus comes on. So the first dotted line is onset of motion stimulus. Second dotted line is the choice reference. And here I've split the data out into our 4 different conditions. So in blue we have low coherence, and in high we have high. Sorry, in blue we have low coherence. In red we have high coherence, and then the line type indicates whether the distance is low or high. So here it's on the left for dopamine. It's not so clear, but then if you move your eyes over to the right to serotonin, it's a pretty striking effect that we see here. And it's tightly locked to the onset of the motion stimulus. And very early on, there's a separation between low and high coherence trials. And this is sort of an opponent manner to what you might expect dopamine doing. So serotonin levels go up if coherence is low. So it's kind of like an uncertainty signal, which fits with the rodent literature, for example, from Zach Mining's group. Now, what's the really cool thing is that we won't just only look at encoding of task variables. We can also actually look at how were people's subjective reports related to these neurotransmitter levels.
How many people are averaged in there?
So these are 3 patients.
And those are averages of averages?
So these are averages of averages. I think, as I was showing to read this morning, what's really cool is that this is averages of averages, but in every single patient you see this separation, which is really exciting. And for these patients, because we were getting the protocol up and running, here we have about 200 trials, and then for the later patients we have 300 in the surgical suite.
So I just want to make sure I understand.
Yeah.
After the motion and the reference, when you got the last 4 seconds, that cyclicity, what is that?
So here, yeah. So next trial is coming on. So there's some jitter, and your reaction time changes. So it's variable when the next trial comes on. But this might be locked to the generation of a choice, or it might reflect some predictive process of the next trial. So that's what we're looking into right now.
So there's a lot of information that's
Yeah.
out of phase
Yeah.
around 3 and 4 seconds.
So now that you explained a second ago about the group of all the subjects, so when I look at it for whatever it is, 4.5 seconds, though, that big, on the serotonin one in particular,
Yeah.
That they've lived there in arbitrary units of the high coherence.
Yeah.
That's very striking. Huh?
Yeah. Yeah, so obviously you can see there's a lot of information in here. And here I'm just locking it all to the start of the trial. But when you start rebaselining it to the different events that happen through a trial, then you start seeing the patterns, also for dopamine that they're
Could
mentioning.
you say one thing about the low coherence condition where in a very early slide,
Yeah.
and I think one slide back where you're showing the behavior in the patients, there's basically it's flat in that
Well,
on the right. The confidence is flat across the low and high conditions.
yeah.
Low
So so these are averages across subjects.
I know,
Yeah.
but if you squint, do you believe, if you mentally squint, do you believe that this means that when it's low and you can't tell, it just never quite reaches a conscious meta knowledge of how I feel about it? It never ever, it doesn't quite make it.
Yeah, there is something
You
about that
suspect
low.
if you parameterize that,
Yeah.
that you might see a transition to what you see in the high.
Probably. I mean, for example,
We had a distribution
yeah.
of
So,
low to high coherence.
so one thing we could do is that every single stimulus is different, right? So, for example, imagine we applied motion filters and tried to quantify the energy in that stimulus. That would be really interesting to look at does the motion energy, because obviously here we just have low and high. How does the motion energy relate to the neural signal? Is there some kind of step-like
Because in your
pattern?
normative data, it was strikingly also absent with a
Yeah.
lot more people.
Yeah. Well,
For the low coherence.
it is there, but
It's very.
but it's very minimal compared to the high coherence. Yeah. And especially given the impact it has on choice performance. I think that's the surprising part. Okay, so now I'm going to move on to the subjective reports, which is also very exciting. So we listed the reports on about a third of the trials. On the left, dopamine, on the right, serotonin, also locked to onset of the trial. And as you can see, if you look at dopamine, is that there's sort of two stages to it. There's a bump when the motion coherence comes on, and then there's another bump a bit later on, which might be related to the choice. So again, what we're doing here is that we're looking at the relationship between neurotransmitter levels across a trial and how confident people said that they were later on. So this is predicting what they're going to say on that trial. And then for serotonin, you kind of see the opposite pattern. Now the pattern gets really interesting if you start splitting it out into correct and error trials. So in green, we have encoding of confidence when your decision turned out to be correct. And then in red, we have encoding of confidence when your decision turned out to be wrong. So what's interesting, if you look at dopamine on the left, when the motion stimulus comes on, there's a separation. So they move away from zero. However, after the reference comes on, and then continuing from 2 seconds to about 4 seconds, they both seem to be going up. So there seems to be a separation and then a ramping up towards some response. The serotonin data is a little bit more noisy, it looks like, but then again, we're seeing some separation between correct and error trials in terms of encoding confidence. And if you look at on the left-hand side, I think this is interesting because this 2017 paper, where they did recordings from monkey midbrain dopaminergic neurons and then looked at the firing rate across time for correct and error trials, what you see there is that you also see a separation between correct and error trials before feedback has arrived or a choice has been made. Yeah. So, oh, sorry. Yeah, so this is so obviously confidence is confounded with the.
Could you repeat that?
Yeah. So Pearl's question was, what does this 33% refer to, and also, what is the overlap between correct and error trials and different stimulus features, and obviously, correct and incorrect trials are confounded with the stimulus features low and high coherence to some extent. And here 33% refers to the amount of trials in which we elicited a report. So they don't make a confidence report on every trial, but they do so. So there's a 33% probability that that trial will require a report. If that makes sense. Okay. So I've introduced you to a novel task which we use to isolate confidence from its component parts. And I've shown that there seems to be a distinct encoding of confidence probability correct in PGACC on both fast and slow timescales. And then this really cool recent work with Reed and Ken and Steve. I've also shown to you that dopamine and serotonin reflect confidence in an opponent manner, which is very fascinating. So now I'm going to move on to sort of the second part. This will be a bit shorter, depending on Reed's line of questioning. But luckily, I think we discussed some of these parts over lunch, so we won't have to do it now. Perhaps.
Don't ask any
Yeah,
questions.
no, it's not. It's not. Questions are good. So I'm going to move on to public confidence. So I've this far considered confidence as a control signal for individual behavior, but confidence also plays an important role in social interaction. And confidence in social interaction is often used to manage influence. So people who are more confident, all else being equal, tend to be perceived as more credible, more persuasive. And in group decision-making, which I've worked on a lot, and which I'm going to focus on now, confidence is used as a reliability signal, if you like. So opinions expressed with higher confidence tend to have a higher impact on the group choice. So if you have a group member who's more confident than someone else, they tend to have a higher impact on the course of action that the group eventually takes. Now, this confidence heuristic, if you like, introduces an interesting communication problem. Because in order for us to combine our opinions in an optimal manner, we have to somehow communicate our confidence in a mutually consistent manner. So what do I mean by that? What I mean is that in an ideal scenario, you and I would use the same mapping from probability correct to our confidence. So that means that if I was a bit more confident than you, that also means that I would be a bit more likely to be correct than you. However, in the real world, people usually have different mappings, right? So here blue is biased towards high confidence, and then as a consequence, on this particular trial, if you like, blue expresses higher confidence than red, but blue is in fact less likely to be correct, so the group might make the wrong choice. And this is just to provide some background for why this might be a problem, common problem. So we know that confidence varies with features such as personality, psychiatric symptoms, professions. So there are studies to show that finance professionals tend to be more confident in terms of self-report. There's also cultural differences, and then there are also gender stereotypes, if you like. So the question is, can people solve this communication problem? Can they somehow align their internal mappings, if you like? So this is not a trivial problem. You need a lot of experience. There's also this recursive theory of mind problem, because I'm trying to adapt to you while you're adapting to me, and so forth. So it's not a trivial problem. So how did we study it? So this is a version of a task developed by Bahadur Barami in 2010, published in Science. So you have two people performing a sensory discrimination task, and it goes like this. So you and I are sitting sort of at right angles to each other. So we can't talk. There's a screen between us, but we communicate through the computer interface. So we see two displays, display 1, display 2. And one of these displays contains a pop-out Gabor patch, and you might be able to see it here in the first display. And what you have to do is decide whether the pop-out was in the first or second display. Okay? And you respond in this way. So if you think it was in the first display, you move a marker to the left of the center. If you think it was in the second display, you move it to the right. And each step away from the center indicates your confidence. So the more you move away from the center, the more confident you are in your choice. So I've made my choice. I went for the first one and said about confidence 4. Then there's the joint decision phase. Then I see my partner's response. So imagine I see Reed's response. He said display 2 was less confident than me. And what we have done in this experiment is that we have implemented this maximum confidence decision rule. So in any given trial, whoever is more confident determines the group decision. And if they're equally confident, we flip a coin. So before moving on to the next trial, people receive feedback. So here, group went with my decision, shown in blue. Group decision was correct because my decision was correct, and my partner's decision was wrong. So we see the feedback. So I see my partner's response, feedback, we move on to the next trial. And we do this for hundreds of trials. So group members have to learn to communicate their confidence in a way that maximizes joint accuracy. So what do we expect to see if people were optimal, if you like? Well, if people had similar levels of expertise, a sort of sample from similar distributions of probability correct, if you like, and they used the same mapping, then we would expect their public confidence to be similar. Yeah? However, if they had different levels of expertise, sample from different distributions of probability correct, then we would expect their public confidence to be different. I guess this makes sense. So confidence should track accuracy. We can also quantify this. So what we have here are simulations of how well a pair of two group members would perform under different confidence levels. And here the two members have similar levels of choice performance. You can see that the sweet spot, as indicated by red, lies along the diagonal. So we should be equally confident. However, if we have different levels of expertise, the sweet spot moves away from the diagonal. So here, member 2 is better than member 1 and should be more confident, right? And we can use these simulations to compare against empirical performance. So what did people do given the differences in performance? That's what I'm showing here. So on the x-axis, we have the difference in accuracy between member 2 and 1. And then on the y-axis, we have the difference in their confidence. And we would expect groups to fall in these two quadrants under optimal performance. And what we see across experiments is that this is roughly the case. So we have a positive slope. However, this slope is very different from what you would expect under the optimal model. So there's this clustering around the 0 point along the y-axis. And this is the term, we describe this kind of behavior as confidence matching. And I think it's nicely illustrated here. So these are groups. On the left, you see data from when they performed the task on their own. On the right, you see data from when they performed the task together. And as you can see, when they're paired and perform the task together, the data points, this is mean confidence, sorry, I should have said that. Mean confidence for member 1 and 2. And you can see when they interact, the data points are drawn towards the diagonal. So there's this matching behavior.
What does the isolation mean there?
So isolation means that you and I are sitting there doing the task on our own. Sort of similar trials, but it's...
I don't see anything you do.
No. So it's just...
You would expect this to tighten up along the y equals x line if all it was extra information, whether or not it was a social person, They'd have more information.
About what you should do?
Yeah.
I guess what...
They see your guess, and they see the percent correct, and they can build a distribution over that.
Yes, but I think what's important, so what we show in a ton of different analyses, is that where you end up depends on who you're working with. So it's not a...
There's an inertial term for another person.
Yeah.
You can't just move them.
No, I know. But what happens, so I guess the point is that the midpoint doesn't, it's not a function of the social task per se. So where we end up, the convergence point depends on where we started. So we always meet in the middle between you and me. So we're not moving towards 3.5, let's say, necessarily.
Okay.
But so I'm not going to go into it here, but we did like a ton of controlled experiments and exactly for these. I know you didn't review the paper, but
I did not review it.
yeah, for these
I
reasons.
will note it got accepted, though.
Yeah, it did get accepted. So miscommunication reduces joint accuracy. Let me explain. So under the optimal, in the optimal scenario, where you and I use the same mapping, we get a nice separation in our public confidence when our probabilities of being correct differ. However, under confidence matching, what we find is that in order to end up with similar distributions over public confidence, then we have to misalign our internal mappings because we start off from different places. Yeah. So the idea here is that confidence matching induces miscommunication, which has an impact on joint accuracy, and that's what I'm showing here.
Or situations of non-cooperation, like a poker game or a negotiation, where your incentives are mismatched anyway.
Yeah.
Right? right? Those lines aren't leaning like this, they're leaning like that. So in other words, instead of trying to cooperate, you emit,
so
faults, you do a pseudo-public confidence, right? Because on purpose.
Yeah, so here,
We have a lot of
so the optimal point here, so I'm going to come to that in the very last part, but here the optimal point is under the assumption that you and I love each other and want to get as many correct choices. Yeah, this is the Danish model.
The American model
Yeah.
would be tilted to the right.
Yeah. Although I should say we did a related task we've done this task in Denmark, England, Iran, and China.
Are there differences?
No. Yeah, so that's this. Well, we obviously thought that there would be.
They love each
Yeah.
other, right?
But either we have created a task which sort of induces democracy, or we have found some kind of universal, if you like. So maybe you guys should try it.
We have a present problem.
Yeah. Okay. So why are people engaging in this behavior? So the different kinds of costs that they could be trying to minimize. So one is social cost. Maybe we match to reduce conflict, diffuse responsibility for difficult decisions. Another kind of cost we might be trying to minimize is a cognitive cost. It's definitely easier for us to match computationally than it is for us to figure out which mapping is Reed using, how should I adjust my mapping, and so forth. And there's also a sense in which matching can be thought of as an equilibrium, because if we end up along the diagonal, we're sort of better off than if we had the worst points along in the landscape, if you like. Okay, so the very last part is okay. So this far, I've talked about private confidence, so these kind of computations and the neural underpinnings. And what I'm going to do in the last 10 minutes or so is that I'm going to present some preliminary results from an ongoing project in which we studied how people adapt their confidence as the social context changes. A bit like what Reed was alluding to. So imagine me interacting with my parents, my wife, colleagues, and friends. You might imagine that my behavior changes for a fixed problem, if you like. And I think it's intriguing that in the first study I presented, prefrontal activity, dorsolateral prefrontal cortex, did not track probability correct, the statistical signature of confidence, if you like. Because many studies, many of them by my supervisor, Steve, have shown that this area is involved in the generation of confidence reports, and it varies with people's ability to introspect or reflect on their internal experiences. But I think a hypothesis that we're entertaining now is that this prefrontal area, instead of being directly involved in the computation of an internal sense of confidence, instead supports the transformation or the mapping from this internal estimate into some context-dependent representation that you can communicate and use for social interaction. Okay, so why would we adapt our confidence to the context? So different social contexts have different reward structures. So sometimes it might pay off for you to be bimodal, being either completely unsure or completely sure. So there's research which suggests that politicians who are not in two minds, but sort of either really sure or really unsure, get more votes. Sometimes it might pay off for you to be submissive. For example, when you're in a clear hierarchy situation, sometimes it might pay off to be dominant, as confidence can deter competitors and so forth. And then only very rarely, perhaps in science, it can pay off to reveal one's true belief. Yeah. But this is just to give you a flavor, an intuition for why you might want to have these strategic dissociations between what you say and how you feel. So how did we study this? So again, we had the dot motion task, and what we have is subjects first see as the sensory stimulus. Now they only have to choose left and right, and we manipulate the coherence. So we have low coherence to high coherence. So sometimes the trials are easy, sometimes they're hard. So you see the stimulus, you make a choice, and then you're shown your partner. So in this trial, I'm interacting with Jennifer, let's say. And the partners vary in terms of their confidence profile, from partners that are very low in confidence to partners that are very high in confidence. So I see the partner, I'm informed about the social context, and then I submit my report. Then I see my partner's report, and the decision made with higher confidence is selected, and we get an outcome depending on which decision was selected. So in this trial, Jennifer's choice was selected. She was more confident than me, but her choice turned out to be wrong. So And I should say also, when we take this to the scanner, that this showdown phase, we don't have it there. So that might become important later. So I'm just going to move along this. But the basic intuition is, as we move along this axis, so imagine this is me, I'm member 2, I'm interacting with different partners. And as the partner's profile changes, then I have to shift my confidence in order to maximize reward.
So just real quick, what's the payment structure look like? Is
So-
it the subject or is it the other person they're playing with?
No, so subjects are told that so they have four partners, and then let's say for each of those partners we're going to pick a set of random trials, and then you get $1 per correct trial. Okay, so and that's the money you receive. Subjects are told that these are previous subjects, which is partly true. So you know they're not adapting to you.
Okay, but the previous subjects they're not getting
No.
the same handbook?
No.
Thanks.
Okay, so what do we see in behavior? Well, we see confidence increases with coherence, which you might expect. And we also see the confidence increases with the partner. So this is data from 30 subjects, here color-coded without error bars. And then we combine these two plots. So you see an interaction. So my confidence is higher when coherence is high, and more so when the partner is a high confidence partner. Okay? So does prefrontal cortex support private-public mappings? Well, what we're looking for is we're looking for some kind of computation which takes a private sense of confidence, adds some social context, generates a public report. We might expect different kinds of activity profile. So you might expect a linear profile increasing with partner confidence, or we might have some kind of U-shaped profile. And under the U-shaped profile, activity might be higher or lower when a partner is very different from you, when they're more towards the extremes. So this kind of profile sort of encodes how much do I have to change from what I would have said in some kind of absolute manner. And if we look at activity, the activity time course, so this is logged relative to the partner reveal to when I see the social context, you can see that prefrontal activity goes up when the partner is the highest confident or the lowest confidence partner. So you see this separation that I introduced you to. Yeah.
That's high and low.
Yeah. So
That's
very high, very low, and then these are the two medium ones at the bottom.
At least a V, it might not be a U. I'll quite high.
No. Yeah.
Okay.
Yeah. So this is the simple story. Yeah. So this is looking at the linear and quadratic term being encoded in the activity time course. There seems to be no linear term. Okay, so what we're currently doing now, and something I'd like to do with Reed and Ken later, again maybe perhaps with voltammetry data, is to look at how people build these context-dependent representations. And what we're doing now is to use our pre-scanned data, where people got feedback to look at how these representations are being built up. And in particular, we're focusing on updates following outcome delivery. Okay, so first of all, subjects seem to track factual outcomes. So what we have here is confidence on the y-axis, and we have the outcome on the previous trial on the x-axis. Green plus is when previous trial was correct, red cross is when previous trial was wrong. And then in yellow, we have the trials where I was selected, and in blue, the trials where you were selected. So when my choice was selected on the previous trial, next trial comes around, then I tend to be more confident if I was correct on the previous trial. And we see the opposite pattern when your choice was selected. So if your choice was selected and you were wrong, then I tend to be more confident next time around. And this kind of makes sense if you think of subjects as trying to find the right level of confidence that would maximize reward. Now, interestingly, there also seems to be a counterfactual component. And here, counterfactual, the counterfactual aspect are outcomes that could have been obtained had subjects chosen a different action. Yeah, so sort of hypothetical or fictive outcomes. And here, please ask me if this is not clear.
What is DOH mean?
Yeah. Cool. Where were you in the 90s? Yeah. Too busy doing actual work.
What is DOH?
It's sort of like let's see if it becomes clear when I explain to you which scenarios
This is, are we parsing on age here?
Yeah. So what we have we're looking at the difference in confidence between different kinds of scenario, and we have the first scenario we're looking at are trials where my choice was selected and my choice was correct. Yeah. Then the alternative outcome could have been obtained if your choice was selected, right? And what we're looking at is difference in confidence on the trials where my choice was selected and you were wrong relative to when you were right. So this is kind of like, it could have gone wrong, if you like. And I tend to be more confident next time around. This is the opposite. So this is when you were selected and you were wrong. However, the outcome could have been positive. So then you tend to be more confident when the outcome could have been positive compared to when it could have been negative. Does it make more sense now?
Yes.
Okay. So the effect is even stronger on the trials where my choice wasn't selected, your choice was selected. So when your choice selected and was correct, then I'm less confident next time around if my choice was wrong. So it could have gone wrong if my choice had been selected. And then we see the opposite pattern. When your choice was selected, your choice was wrong, but we could have gotten it right if my choice had been selected, yeah? So these are sort of counterfactual components that go into the updating. And what we're doing now, what I'm working on with a Ph.D. student who's visiting our group from Iran, Sarah, is to take these components, counterfactual and factual components, and formalize it in a reinforcement learning framework, building on work that Reed and Ken and Terry have done. But sort of it's a little bit more complicated because you have this private-public space that you're operating over. Okay, so I have two slides left. Okay, so now I'll just talk about contextual modulation of social behavior more generally. And I think we can all agree that successful social experiences require sensitivity to context. So what is appropriate in one context may be totally inappropriate in another context, and what we think or feel may clash with the behavior that's required of us. So for example, it might be appropriate to express gratitude for a present you don't want, or it might be appropriate to decline food even though you're hungry. And it might also be appropriate to refrain from sharing some joke that you really love. Might just not be appropriate for that situation. And I think the importance of such context sensitivity is perhaps most apparent when it's lacking or not quite right. So this is the case in many psychiatric and neurological conditions. It's also the case for some people who might not have been diagnosed, but have it as a trait that they struggle with context. And this can cause great distress for the individual because you get negative outcomes, and for the family and friends who are affected by it. So I think one useful way to think about contextual modulation of social behavior is in terms of private-public mappings. So the scenarios I just discussed, they all involve some kind of filtering or some kind of transformation from a private state to a public state. And I think confidence is a useful model system for studying these kind of problems. And on this account, we can sort of think of contextual modulation of social behavior as involving a set of sub-problems that we have to solve in order to produce a context-appropriate response. So these are just some of them. So for example, you have to know what context you're in, so you have to recognize the context. Once you've done that, you have to figure out what you should do. So which of the many actions you could take should you take? And once you've taken an action, experienced some outcome, you have to learn something from that situation. So you should ask yourself, what should I do next time around? Now what we have is that we have the computational tools for characterizing how people might solve these soft problems involved in generating context-appropriate behavior. But we haven't yet applied these tools to study contextual modulation of social behavior. So I've just added some names here, a bunch of different kinds of algorithmic approaches, if you like. And we also have an idea about which areas might be involved in generating and implementing the solutions to these subtasks. We also have an idea about how these areas might interact. So for example, hippocampus might be involved in identifying the context, which then together with prefrontal cortex is used to trigger some kind of action initiation, select some action, and then striatum might be involved in sort of tweaking the parameters in this process. And I also think that this particular account offers a way of thinking about different routes to social dysfunction. So, for example, an individual might struggle recognizing the context because producing a context-appropriate behavior because they haven't learned the full space of context or because they can't identify the current context. They might be able to identify the context, but they might not be able to inhibit prepotent actions. Or it might also be that they learn the wrong things from social experience. So I just talked to you about factual and counterfactual outcomes. We know that dopamine and serotonin are involved in signaling factual and counterfactual outcomes. And it might be that individual differences or also signaling in various disease states impact on what we learn from social experience. Okay, so to recap, so I've proposed that we might think of a particular private-public mapping as a kind of computational phenotype that we can explore further. And I've talked about these possible interactions between hippocampus, striatum, and prefrontal cortex, and how these interactions might implement different solutions in order to produce context-appropriate behavior. So at the very end, I'd just like to thank Reed for inviting me, Ken, Jason, Terry for working on the voltammetry data I presented to you. So this is incredibly exciting work that I think is going to be very important and a game changer for the field. And I'd also like to thank my brilliant supervisor, Steve Fleming, my postdoc supervisor, and my Ph.D. supervisor, Chris Summerfield, who's at Oxford and Google DeepMind. Okay, so thank you.
By the way, Chris' name said Christ.
Pretty funny. I can't help it when I'm here.
Yeah, it's just
yeah.
Well, thanks.
Christ, please.
We're running a little late, but nobody has questions. You're going to stand there. So here's a rule that I'm implementing called Montague rule. There's always a mercy question for speakers, right? I actually have questions, but
yeah.
Throw that out to the students. Surely you have a question.
I get a little confused with the voltammetry stuff.
Yep.
It seemed like you were plotting confidence levels over time, and I was wondering if I had that wrong.
Yeah. So it's So that's down to my poor explanation. But let me just go back to that. Let's look here. So the way these analyses work is that so imagine we're sampling the neurotransmitter levels every 100 milliseconds. So in a trial which takes about 6 seconds, you'll have 60 samples. Is that right? Yeah,
Right now.
that is right now. And then what we do is that we know on this trial, and let's say I know you said high confidence, 6 out of 6. And what I do is I ask across trials, how does activity or neurotransmitter levels at this time point, does neurotransmitter levels at this time point predict what you say? And then ask the same question at the next time point and so forth. I don't know if this kind of analysis makes...
The vertical axis are
Yeah.
regression
Coefficients,
phase.
yeah.
They're coefficients from a general linear
Yeah.
regression on just the two variables that you mentioned. So each time point is being used to predict what's the report's going to be.
So imagine, so I have all the trials from trial 1 to 60, let's say. And then I have my neurotransmitter time series, and I have 60 of those. Then what I do for each time point is that I just run a linear regression predicting my confidence vector using the vector at that particular time point, and then I just do that through time. So that you can think of a kind of like encoding of confidence in this
the
case.
assumption that the transmitter fluctuation would happen first.
Yeah.
Right. It's not magic. This is not magic.
Yeah.
So
Thank you. Great talk. I have a question going back to the in and out of scanner doing the moving dots task.
Yeah.
An experimental design question. So how important is it that what the person sees is like matched between them being outside of the scanner versus inside the scanner? Like I've done the task myself, and I noticed like even slight differences in like the amount of the visual field taken up by the circle itself can change the difficulties.
Yes. So you're absolutely right that that there is variation as we move from outside to inside the scanner, because outside the scanner we're using a screen, then suddenly we have to use a projector, and all kinds of problems which we have tried to minimize, if you like. Now, I think the important point is that it's very much a within-subject design. So you're looking at the effect of these factors on behavior within that subject, and then we average that across subjects.
So then the kind of calibration about where their confidence, I guess that metric that you calculate, that isn't effective.
I see your question. Yes, so what we did there is that it does predict what is said inside the scanner. So if you actually, because with the computational model, I create a report on each trial, and then I can see how well it predicts what people said on those trials were actually elicited in the report, and it does predict it. But you're absolutely right that there's no guarantee that the weight of the different stimulus factors, the way that their influence and confidence is preserved as you move from outside to inside the scanner. Yeah.
I have a question. Can you go back to the high-low coherence distance, like way at the beginning with the normative group?
With the human, that's not, they're both humans, but the FMI, I meant FMI.
The behavior?
Yeah.
Yeah. Where, right there, yeah. I just want to get you to speculate. So I just want to point out for the students, too, that I think what's good about these kinds of experiments is, first of all, this dot task, this coherence of dots task, is well trodden. This has been used for 50 years in visual neuroscience, both in primate psychophysics, non-human primates and human primate psychophysics. And so it's well understood. People have played with it in all kinds of ways, and it's used to look at the way you integrate information to make decisions about it. And then go look for neural correlates of it, go poking around with electrodes. So, but I think what Dan and Steve's work has done is transport this to two really interesting domains. What do I know that I know, and how do I feel about it? Okay, so those seem ineffable in a way, right? And so he's taking a very concrete task that's used to sort of pump information into your nervous system and study the nervous system in the sense that its job is to take apart the world and then put it back together again. That's changed. Now the world is Bayesian, so the brain comes with a lot of models of the world, and they've taken this task and asked these two questions, which would have been something people argue in a room about with a lot of words, and turned them into experiments. So I just want to do a kind of commendation round on that. And the second thing here is, this is a very interesting effect to me, because somewhere between whatever that coherence level is and that coherence level is, starts to tip. I don't know if that would pass between these two ends. Do you know?
Well,
It's as though you don't know.
Yeah.
In other words, at low coherence, as you go from hard to easy.
Although, bear in mind, you are more confident than you should have been objectively.
Yeah.
But
it's not a clean signal.
No.
So you guys are looking for a signal here, and so that's an interesting gap to try to fill in in future work. So, is that how do you guys talk about it under the covers? I mean,
so here, I mean, there's so both
because it looks horizontal.
Yeah.
You said squint. What is that? It's a horizontal line.
No, but it is increasing.
Sort of.
If you zoom in, you'll see
okay,
it.
Compute
But I think
compute the delta increase as a fraction of the increase in the red.
Yeah. So what I didn't show here, and what we actually didn't include in the paper, is that we were quite interested in sort of oversensitivity to coherence, to sensory reliability, and what we actually did is to use the slope, as seen here, as some kind of proxy of whether people over or underweight sensory
The
reliability.
slope for each person.
Yeah.
Yeah. So I kind of
So
like,
we-
I was thinking that.
yeah.
I was just thinking there must be people,
Yeah.
and the problem feels easy.
Yeah.
They generate this big confidence
Yeah.
signal, and people that are a little more parametric,
Yeah.
and how those would sort. So.
Yeah. So that was sort of a panel 4 in some figure that went.
Okay.
So,
the reason
but-
I think it's
yeah.
important for future experiments that you encounter planning is that these methods that are used in the striate, these are all in striatum and the caudate nucleus, these recordings of dopamine and serotonin.
that method has now been transported to wires. Standard commercially available electrophysiological wires used in people when you're monitoring for epilepsy for various reasons, where they put in 10, 20, 30 wires into your head with anywhere from 200 to 400 contacts. Okay? Or in a rodent experiment where you have a lot of access in your recording. Okay, so that means in a human being, you could go back in with a lot of this behavioral data and look at a person who's being monitored for epilepsy, who obviously has seizure disorder, but every chunk of their brain is not ill and seizing, and make these measurements from all over the place.
Yeah, I think this might be some kind of measure relating to awareness or sort of.
Right. So you might see that when you record it from enough sites and have a measure like a neurocomputational measure.
I mean, I guess like Standerhin thinks of it as this kind of ignition.
That's kind of blunt, but okay. So, anybody have one more question? Yeah.
Have you experimentally manipulated dopamine, for example? Do you have any data or any intuition about whether that would constrain private confidence or public confidence, or the two differently?
I mean, so I know people have looked at. So the question is whether, I guess, drugs, if you give people L-Dopa, if that affects their confidence levels. I'm not aware of literature that have looked at that or know. Or maybe that's actually wrong.
It changes your it changes your risk propensity on gambling games.
Yeah. But specifically in relation to confidence, I think there's no impact of.
Nobody's ever done that with best.
Yeah. So no one's. I mean, I think you would rather go and measure dopamine levels in the striatum.
Parkinson's patients who take
Yeah.
dopamine precursors,
Yeah.
they suffer from overconfidence. They suffer from this reckless confidence syndrome that people talk about. And this sort of migrates into something that's hard to distinguish from lack of inhibition, like gambling behavior, eating behavior, stuff that they didn't do before they were on the L-Dopa drugs.
But as we discussed, it's quite hard to tease apart that impulsivity and so forth from confidence per se. If that's sort of what you. There's
Do
definitely.
you have any sense whether it would have a differential effect on the private confidence or the public?
I mean, given this, I would say yes, but I seem to, in the back of my mind, to remember that a colleague of mine, Tobias Hauser, had a paper in eLife last year, I think, looking at metacognition. And I think it was a drug study looking at norepinephrine and dopamine, and they didn't find an effect of dopamine drugs on confidence. I seem to remember. So it might more be related to.
I suspect that it does, though.
Yeah.
They just haven't had a way to measure it. The effects are fast and they adapt. And I just think there hasn't been a method to.
But I guess I mean, for example, and I don't know if you think this is, but are there differences in baseline concentrations? Is that informative?
Who knows?
Yeah.
I'm not.
So that might be one way of looking at it. Yeah.
Hey, thanks again for coming. We appreciate
Yeah.
you.
Thank you. Thank you.
That was great. Well, thanks for going over so much stuff, too.
Yeah.
And I was sort of,
No, I know.
my questions were
No, I know.
the audience, right? So these students are they're from very wide-ranging backgrounds.
No, but I think it's really useful when people do that, because people sometimes don't do that. They might understand, but not. Because people won't ask.
It's good. But Chris should be demoted in Christ.